All right, everyone, welcome. I'm going to go ahead and kick us off with some intros. My name is Jenna Gallegos. I'm from Samba Scientific, and I will be your moderator today. Our webinar today is sponsored by N6 and includes a great lineup of speakers. We have Jeremy Wilkinson, who is the global lead and microbial genomics lead at PacBio. Mary Arrastia, who is a senior field application scientist at N6. So Jeremy and Mary are going to give us tech intros. And then we have Dr. Charles Mason, a research biologist at USDA-ARS. In our webinar today, you're going to learn how the USDA is leveraging long-read sequencing and ICON PCR technology in order to dramatically increase unique ASV detection in complex microbial communities. Without further ado, I will go ahead and pass it over to our first speaker, Jeremy.
Thank you. Okay, so I'm going to be giving just an introduction to full-length amplicon sequencing for superior microbiome taxonomic resolution with PacBio and also with ICON PCR. So what PacBio sequencing is, it's more complete and accurate long-read sequencing through our two main platforms. So the Vega, which is the benchtop system there on the left, and then the Revio, which is the higher throughput instrument on the right. And this can accompany many microbial genomics applications, including shotgun metagenomics, microbial isolate assembly. Epigenetics come natively with whole genome sequencing applications if you don't use an amplicon-based approach. Viral sequencing through a variety of different approaches, targeted sequencing, and then what our focus is today is full-length amplicon sequencing, which can be 16S or ITS or 18S, et cetera. And so how this is accomplished, PacBio hi-fi sequencing, again, has highly accurate long reads, and this is through circularizing our DNA in the library prep process with what we call smart bell adapters. And then the polymerase actually sequences around this circularized DNA molecule multiple times. So it's these passes or consensus sequences that are able to correct for these random errors that are these little red dots that you can see in this plot. And what this ends up giving is a consensus sequence, which is about 99.9% in accuracy on average, but typically we'll see quality scores of Q40 or even Q50 for full-length 16S, for example. We have lots of data sets available through publications on NCBI, hundreds of them now. The mean relinks that you can get from these data sets are between 5 kb and about 20 kb, but for shorter amplicons, they can be smaller. And then we also have lots of data sets available on our website if you want to check those. So we have many library prep solutions for microbiology applications. So this starts with Kinnex, which you'll hear some about from Charlie later, which is a concatenation approach to concatenate anything that is an amplicon or cDNA into a longer molecule. So for example, a 16S, it's a 1.5 kilobase region. And so what it does is it concatenates 12 of those molecules together to make a 18 kb insert library. So you take full advantage of PacBio hi-fi sequencing. And then what that does is it increases the number of reads that you can get per sample and or the number of samples that you can sequence on a SMRT cell. We also have other partners like Zymo, who has a quick 16S full-length library prep kit that can set up front and have the PCR reagents for you. And then like today, what we're talking about is N6 with the ICON PCR, which is really useful for controlling things like overamplification and chimeras through their autonormalization. And then we have lots of different library prep options for whole genome sequencing, but we're not going to touch on that today, including another partner that's just Sequel with the Longplex multiplexing kit, which is really good for the lowest cost and the highest throughput capabilities. So just focusing on amplicon-based microbiome sequencing, we know that bacterial species are highly similar. They're difficult to distinguish with partial 16S sequencing with short reads. And so this is really a paramount study that came out back in 2019 from George Weinstock's lab at Jackson Laboratories. And this is showing the cultured isolates of the healthy human gut. And those are shown in this dendrogram here on the left. Each one of these branches are the cultured isolate, and each one of the colors of these branches represent a bacterial family, which the legend is here on the bottom. The x-axis are the variable regions of 16S, so 1 through 9. And then the shaded regions in this plot are areas where SNPs are occurring. So SNPs occurring between these isolates. And so if you were to sequence with just standard V4 short read 16S sequencing for two of these really important human gut bacterial families. So gray is Bifidobacteriaceae, and then the pink here is Enterobacteriaceae. What you see in this box is nothing. So there are no SNPs that are occurring for those two families, which is V4 sequencing. So you cannot distinguish them past the family level. However, if you expand that to V3 and V4, which a lot of folks have done recently, which is great, it does give you some added resolution. You start to see some SNPs pop up, but still very few. However, if you expand across the entire 16S gene, you start to see SNPs occurring in lots of different variable regions. So this gives you the capability now to distinguish these individual isolates for these two bacterial families down to the species or strain level. And so Charlie will talk more about this project later, but this is really showcasing how you can get the best in class for both quality and costs for 16S sequencing by using PacBio with Kinnex. So in this study for Mediterranean fruit flies, they're able to uncover potentially unique strains and greater diversity than they initially thought in these library populations. They were able to uncover strain level diversity that was not immediately obvious using their former approaches. And so through implementation of Kinnex 16S on Revio, the cost was actually on par or lower than their 600 cycle based short read approach. And then you can also go beyond just sequencing the single gene. So you can actually sequence the entire operons of either prokaryotes or eukaryotes. And so this example paper was published last year. It came out of the folks from APC Microbiome, as well as Chagas in Ireland, where they were evaluating the long read platforms for sequencing 16S, ITS, and 23S, which is a 4.5 kb region. And they found that PacBio has the highest quality data. It has the capability to generate OTUs with 99.9% similarity, which we know is strain level. ONT, on the other hand, required additional advancements to produce data with sufficient accuracy to provide strain level results or even OTUs at only 97% similarity, which we typically would classify at the species level. So they said that PacBio's higher quality has currently made it the more suitable choice for getting strain level information. Similarly, this is done in micro-eukaryotes, so 18S, ITS, and 28S. So again, a 4.5 kb insert where they compared across long read technologies in this microeukaryotic mock community they created. PacBio was able to provide the best coverage of this mock community, getting 40 of the 42 taxa detected, whereas other long reads were able to get 21 or 32 respectively, and also incorporated UMIs. They also found that PacBio demanded the less labor and showcased better robustness and reproducibility of the workflow. The other advantage is that with our systems, you get the consensus sequences like I talked about on the first slide directly off the machine. So this saves a lot of time on the bioinformatics processing. So that advantage combined with a much lower cost per consensus sequence positions PacBio as the current method of choice. And then lastly, there are some issues that may pop up when you're amplifying longer fragments like this. So even 16S 1.5 kb, but especially when you're amplifying the 4.5 kb region that I just talked about, you can have issues with overamplification, especially chimeras. So that's where Charlie talked about this again later. This is another one of their papers where they combined N6 icon PCR with PacBio Kinnex Full Length 16S on 7 agriculturally relevant sample types. They found that autonormalized reactions consistently retained a higher proportion of sequences following denoising and chimera removal, exhibited fewer substitution-based errors, and produced more even redistributions across the samples. So this enhanced both data quality and lab efficiency, particularly for the production scale microbiome sequencing. And then lastly, I just want to give a plug here. We do have a ongoing SMART grant specifically for metagenomics. So this is shotgun metagenomics, so whole genome sequencing, not 16S. So if you are doing some work in shotgun metagenomics, please do check this out and apply. You can find it on our website if you just search PacBio and SMART grant. And this is also co-sponsored by Seek Center, who is also an owner and customer of N6 Icon PCR. They also use this for their full length 16S and their Connect 16S services. And that's it for me. Thanks for your time. I'm going to hand it over to Mary now.
Thank you, Jeremy. And today I'll be talking to you about icon PCR, basically a new way to think about PCR amplification in your library prep workflows. Next slide, please. Awesome. So let's start with where the field is right now. So genomics is now operating at a high throughput scale, generating massive data sets across a variety of different fields, from population level studies to single cells, plants, and microbiomes. And with the advancement of AI and machine learning pipelines, this raises the bar even further because these models are only as good as the data that's going in. The problem is that many genomics tools are still running on legacy equipment, where these tools not only slow us down, but they could quietly raise costs and slow and compromise data qualities that are not always obvious until you're deep into the workflow. So as a result, we need to develop novel tools to keep up with the pace and scale of genomics research. Next slide, please. So one of those older tools that has seen improvements in today's genomics research is sequencing. So for the past decade or so, genomics was previously hampered by the cost of sequencing as they were quite low throughput. But as tools have gotten better, both in throughput and efficiency, the cost of genomics workflows have shifted quite a bit. Where these days, the major cost associated with genomics is not sequencing, it's actually now library prep. Next slide. So many of you might be familiar with this particular workflow as you might be running library prep on a fairly regular basis. There's many steps involved, from sample quant all the way to library normalization. And on the next slide, really, the key thing that's only essential in library prep is the adapter addition. Really, all the other steps outside of that are manual, they're repetitive, and they add quite a bit of variability. None of them really improve the biology or the data quality itself. Really, they just add time, cost, and increase the risk of failure. Next slide. So why is this the case? Well, all of these variable and consuming steps of the library prep can be tied into one key step, which is PCR amplification. Because PCR yield is unknown going in, you're forced into upfront quantifications and normalizations to control for the PCR cycle number dictated by your protocols. On the back end, you may not know what comes out of PCR until you actually do the library cleanups and the quantifications. And if you're going in with a sample that's of a different type or a different quality in comparison to what your protocol recommends, you won't know until it's too late. The result of that is what we summarize as garbage in, garbage out, where bad PCR runs can equal failed runs and also bad data. Next slide. And for the longest time, the field has been solving the wrong problem. When it comes to focusing on good PCR, most of the time the field has been focused on getting sufficient libraries to achieve normalization for the sake of read balancing and sequencing. But if you over-amplify your library, for instance, normalizing for read balancing will just carry over PCR duplicates and chimera products that were formed during PCR. But when it comes to achieving true normalization, it's the combination of the two things together, read balancing plus optimal amplification. And achieving optimal amplification, which is getting every sample to the right amount of product without them being under or over amplified, is still the true issue today. And many conventional tools don't really address this issue. Next slide. So in terms of PCR, this is generally the fundamental design. Every thermocycler on the market only lets you set one thing, which is the number of cycles. Basically, you're hoping that the number that is chosen will land you in the right spot along the PCR amplification curve, but you have zero visibility as to where you actually land. You don't really get to see how much product is being formed in the process. But with the development of N6 as a company and the creation of the icon PCR, we can now flip that axis. So instead of having to guess cycle number going in, you get to control the amount of PCR product that's actually made. And basically in icon PCR, if you tell the target, the wells will basically stop once that target is reached. Next slide. In order to achieve that, that really requires a real hardware shift. ICON PCR is the world's first thermocycler with individually controlled wells, which is a big contrast from previous thermocyclers that rely on a solid metal block to control all wells of the reaction. With ICON PCR, we have two instruments to support the individual control of wells in our ICON 96 and ICON 16 format, allowing you to run up to 96 individual wells in in every PCR reaction. Each well has real-time temperature and fluorescence monitoring, which means every well can be controlled independently in real time. So if one well is being held at 4 because it's hit the right amount of target, but the surrounding wells need a few more cycles to achieve that yield, the other wells around it can be ramping up to their next step of cycling while a well is held at 4C. So overall, the result is a system that's built on what we call respect for the read, where every well gets exactly the right amplification it needs. Next slide. Next slide, please. Thank you. So in practice, the instrument already works with what you're already running. It uses standard PCR plates and strips, accommodates between 10 to 100 microliters of reaction volumes, and uses your existing master mix reagents and protocols. All you have to do is spike in a little bit of green fluorescence dye, such as cyber green, to monitor your PCR in real time. The feature in the instruments that makes it unique is what we call autonorm. Basically, autonorm knows when to stop your samples at exactly the right time, ensuring the good quality samples don't over amplify, but lagging or lower quality samples have the time it needs to reach its ideal endpoint. We've demonstrated its capabilities across a wide variety of library prep assays, and they've been applied to various sequencers, and the instruments today are made to be compatible with your existing automation tools. Next slide. So this old workflow can now be, we can take this and improve it so it becomes, next slide, this new and simplified workflow with ICON PCR. So because AutoNorm knows when to end your samples at exactly the right time, it allows you to skip the upfront quantification steps that are typically required to normalize the input before going into PCR. On the back end, all samples are amplified and normalized to the same level, which means you can pull all your samples together and perform an individual cleanup and quantification before sequencing, which dramatically simplifies the process. Next slide. So we applied ICON PCR to a variety of different applications, including metagenomic assays. Here we carried out a 16S kitchen sink experiment where we combined a diverse set of different sample types, including soil, fecal, skin, and so forth, with inputs spanning over 6 orders of magnitude. Under normal circumstances, you wouldn't mix such a wide variety of samples, but ICON PCR can easily carry this out in a single run. AutoNorm handled the normalization, and you can see that the read count is balanced across a wide variety of different samples from this run alone, with CVs as low as 15 to 20% per sample type. Next slide. In this other microbiome application, we prepared full-length 16S ribosomal RNA libraries from soil samples, and then these were sequenced using the PacBio Sequel II platform. Overall, ICON PCR improved the richness and the taxonomic depth associated with these samples, even detecting several microbial classes that were not previously seen in comparison to standard approaches. Furthermore, ICON PCR with AutoNorm significantly reduced the chimera and the PCR duplicate rate, allowing for more unique reads to come through and revealing more information than what was previously seen. Next slide. And while here we showed applications related to 16S or microbial studies, we've applied ICON PCR to a whole host of applications, including RNA-seq, FFPE samples, single cell, and so forth. We have full-length application notes on our website if you want to learn more and see the power of ICON PCR with AutoNorm across these different fields. Next slide. And if you don't take my word for it, all experts in the genomics field, including today's speaker, will tell you the same thing, that PCR will make or break your data quality. And there's tons of publications highlighting it. where basically the key message is controlling PCR is universally crucial. And this is why we now have icon PCR with AutoNorm. Next slide. Thank you so much for your time today, and we would love to hear any questions about your work and talk more about your research and introduce where AutoNorm can help. And now I will pass it off to Dr. Charles Mason, where he'll reveal his work, reveal more about his work. Thank you.
Good morning, good afternoon, aloha. Thank you all for the warm intro. Thank you all for introducing me and for kind of giving me a nice takeoff lane to be able to talk about what we're going to be, how we're implementing these techniques here in ARS. A lot of what we do in ARS is considering we want to work on and improve crop production and crop health. And this is a coffee farm in Kona, Hawaii. And what we're really interested in is how we can apply agricultural microbiomes to improve crop health and crop productivity. Part of that is managing pests and pathogens, and my lab in particular works quite heavily on how we can utilize these approaches to manage different types of organisms. Specifically, we work quite a bit with agricultural insects. These are basically we work on different types of species, that have different microbial associations. And what we do know about different insects is that oftentimes they exhibit variable compositions and titers, as in these different insects have different sizes, and then that their microbial biomass may vary quite considerably between each three of them. Some of these associations, at least for some species, are unable to be resolved using partial 16S hypervariable reasons, much like what Jeremy mentioned earlier, and what I'll talk about in a little bit. Yeah, and again, we exhibit different titers and compositions. And so when we were approaching and thinking about amplicon sequencing, we were really interested in how long-read 16S, in particular, the kinase concatenations strategies, can really help us inform our systems and some of the goals that we're trying to implement. And some of the appealing options are that long read is going to provide more information than 16S read than the partial 16S reads that are obtained by short read sequencing. That we expect that these amplicon-based methods would be transferable across samples, that they would be scalable, so we'd be able to go from either doing a set of a dozen to hundreds. And then I think in particular, and like when the Kinix platform came online, this really allows us to approach the sequencing with a greater degree of depth, and allows us to sequence not just a dozen, but upwards of 300, 400 samples at a time and get considerable depth and be able to make ecological inferences. But with this, there are some limitations that we initially considered when we were making these approaches. And in initial testing, we did identify that conventional PCR was going to introduces errors, and those errors manifest in kind of results in sequence loss. There's also this aspect of intragenomic variation in the 16S. Certain species that have 4 or 5 copies, they might all be the same, but in other cases, those 4 or 5 copies may exhibit may differ in different ratios. And then, a comment that we had at the beginning of our studies is like, well, is this really going to be better than partial 16S? And I think at this point, hopefully by the end of this webinar, that you're going to agree that it is better than just partial 16S at costs that are comparable. So to minimize the error rate issue or the residual error rate issue, this is where the icon PCR and autonormalization techniques have really allowed us to streamline our approaches and really minimize those issues. Again, we expect that when we first started utilizing this approach, we were like, well, this is probably going to hopefully will reduce some of the residual error that occurs during PCR and prevents the over-amplification of samples. And that will improve our data quality, and we'll have more reads retained at the end. And then as an advantage, so we have we're not a core. But we have multiple labs who are utilizing these approaches in our location. And this really appealed to us because it aids in streamlined library preparation, where anybody can come up to the instrument, be able to set up a PCR in a 96-well plate and put it on the machine, and then be able to make decisions based off of the data to how their library is performing and how their library can be, whether their library can be pooled. And so this really allows us to have not just a single user, but have upwards of 6, 7, 8 people in our location be able to utilize the instrument reliably and have consistent results between different users. And I'm starting to talk about some experiments that we did to really kind of look at how all these different pieces can be integrated and whether they are not better. And I'm pleased to say that this paper was published online yesterday. So if you want to know about the details of the methods or some of the figures I'm not talking about, they're all present in that paper, and it is now available online and peer reviewed. So this is a case study that we implemented where we took different environmental samples and we basically conducted them to what we call a series of different library pools. So on the left, we have basically three different library treatments. One is which we use all these were performed on the ICON instrument, but under different conditions. The first was autonormalized and then just blindly pooled. The second we ran for 30 cycles and 24 cycles. And then we did a magnetic bead, we cubic quantified each individual reaction, and then we pooled them in equal molar concentrations. There was, of course, some that were potentially under amplified, and so those ones would just got the entire thing pooled. And then we had a set of 20 cycles, which was pooled blind. These all get went into the same Kinnex PCR concatenation and library prep. These were all the initial PCRs were barcoded differently, and this enabled them to all be pooled together and basically couldn't sequence on a single PacBio Revio smart cell. We used Lima implemented on SmartLink for desegmentation and demultiplexing, and we did these under a strict platform where we basically want to have 100% identity between barcodes. And then using our HPC cluster, we basically did quality screening using Q-scores, we trim primers, and we used Data2 for denoising and chimera removal. The set of samples we used were selected with some intention. We used five different six different arthropod species, sorry, and which represent different host ranges, different taxa, presumably different microbial biodiversity in different titers. And then we also used a soil sample and a leaf sample. And the reason that we're utilizing these different sources is that we sometimes have libraries that are kind of encompassing from soil to plant to insect that we want to analyze under the same cell. We implemented positive controls. We implement positive control and we use the Zymo Biomics DNA and extraction standards as our positive control, and we modify the reference to support some of the hi-fi reads that we get. But we utilize this on all of our approaches just to monitor what kind of residual error rates we're receiving and whether or not we should have an expected, like on the icon PCR, we're able to monitor this is where this standard should be amplifying, and we use that as just an internal control to make sure that yes, we're getting continuously consistent results between runs. This is how the amplification of the three different treatments to these different libraries went. You can see under 20 cycles, we have some that are amplified, but some that are under amplified. At 24, we start seeing some potential over amplification of some sample types, but under amplification of others. And at 30, we see that all these samples have amplified, some exceptionally over amplified. And this is what we normally see with the autonormalization approach, is we usually see like it usually tops out around 5,000 RFUs. And it's relatively consistent between different types of samples and different types of approaches. So ultimately, how does this actually process our yields? So we basically had to have a series of quality filtering steps. I'm not going to go into excruciating detail with these, but I want to just highlight at the end, once we get through all of our filtering steps, our 30 cycle PCR library only had about 30% sequences remaining after the end of after the end of this process, whereas our autonormalized library had about 50%. And this is usually, at least on our hands, usually what we expect out of a short read SBS-based library. When we actually look at how the library is distributed, is that we see with the 20 cycles, we see lots of samples that are under amplified, 24 cycles more, there's again more variation and less under amplification. And then the 30 cycles, this is again log scale, where we do get this, it is a pretty dramatic drop off. But the auto normalized is very consistent within a pretty small margin of error. When we actually look at whether or not there's any sort of read variation between within our normalization approaches, whether we look at this at max fluorescence or our stop cycle, so the stop cycles we see, depending no matter when they stopped, we're retaining very comparable numbers of reads despite them being highly different samples. We also see fewer chimeric reads, especially compared to higher cycles in our positive controls. And at least in our hands, our residual error rates have been below 0.05 or 0.07 using autonormalization across all the runs we've used. And so we really are minimizing and mitigating those error rates, especially compared to those that are highly amplified, while also minimizing the number of non-chimeric sequences. But what is particularly, I think, powerful within this, like looking at a variety of samples and looking at it under these different conditions, is that we actually started seeing that there's sample-specific impacts of the PCR protocols. And so this is an example of like using three different samples, that just a highlight of three different samples where we use medfly, pickleworm, and soil. So 2 arthropods in soil. I just want to highlight the soil part, 30 cycles. These were primarily lost as denoised or chimeric reads. And so despite them having comparable percentages retained at our initial filtering steps, there's just higher rates of chimeric formation when samples are overamplified. And this likely there's two things that might relate to this. One is that soil has more microbial biodiversity, more microbial biomass, but also potentially more diversity. And specifically, we do see that there is a negative relationship between sample biodiversity. So the more things that are in a sample and how many reads are retained. So the higher amounts of, and this is all mitigated through autonormalization approaches where we retain significantly more samples, even in higher diversity and higher biomass samples through this library preparation stage. And again, we've used this across, I think 8 or 9 different library preps, all for Kinnex at this point. And these are typical plots that we receive. We usually are obtaining very even sample distribution coming off the instrument. But I do want to make a caveat, at least in our experience, when we encounter samples with exceptionally high DNA concentrations, whether that is isolate based or sometimes sample based, we do have to dilute those samples before we implement them into the ICON PCR, just as a way of being able to, just based on what we expect to be inhibitory processes of DNA not allowing the polymerase to affect the high fidelity polymerase to work effectively. But I think what comes out of all that, what really came out with this is that, and I think Jeremy mentioned this earlier, is that we are able to resolve intragenomic variability in the 16S reliably. So even though there's different levels of loss and error rates amongst the different treatments, is that we are routinely able to still pull out from these hi-fi reads the intragenomic signature in our controls. And so that has had us excited in a way, like when we revisit some samples that had relatively low biomass or relatively low biodiversity, and whether or not there's actually more there. So I want to turn the table to applying this to some ecological questions. This is medfly. We've been working on this for the last couple of years, and we're really interested in the medfly microbiome as a way of improving sterile insect technique and releases that happen in California and Florida. And one of the key questions we've had is, well, how does the MedFly microbiome vary across space and time? And previously, I collected a study where we collected this big temporal data set where we were collecting flies across a nine month period. And I do want to highlight that these data are in a preprint that if you're interested in looking at its cryptic insect microbiome compositions. So this is across a 9-month period, and we used conventional short read V4 based sequencing. And what we identified was that, like when we took all the ASVs that we collected, it was dominated by one. One Klebsiella ASV was present across all these different time points and all these different cohorts and all these different areas. And we're like, well, that's not exciting, but that's the data. So we're going to publish. We ended up publishing it, and it's like, this is just not a particularly exciting data set. Nothing more to see here. But after we started utilizing these Kinnex platforms, we decided to go back and revisit that data set and sequence the same DNA again using Kinnex. And what was exciting to me was that, well, there's actually a lot more there. So we went from this one ASV that was collapsing a lot of the microbial biodiversity, and then utilizing this, we find that there's actually, well, it's maybe not Klebsiella, maybe it's Enterobacter, and that there are a lot more ASVs that are present in the system than what we initially thought. These are V4 on the left, folding 16S on the right, same data sets, same samples, and we get a lot more. We could see that we actually get more separation, we get more structure, we see more variability in these communities than what was only observed using a partial fragment, highlighting the case that, yeah, we get more information and there is more biodiversity in these systems than what was initially obtained using just a partial fragment. So then a key point is, well, does this relate to, is this all noise? Does this relate to anything that's like, this is all intragenomic? This is likely contributing. There is likely intragenomic variability that's driving this. So this is a co-occurrence network, and we use module selection. So we basically took these ASVs, put them in different modules, and these different modules are differently distributed across these cohorts. This also led us to conduct some shotgun metagenomic sequencing using short reads. Problem with that, we weren't able to pull out or map to specific 16S because of limitations on the short read shotgun sequencing front. But what we were able to do is relate, well, how we were able to create mags and then look at, well, how did these relative abundances of these different module groups relate to those mags? And what's neat and exciting to us is that the 16S data seems to be mapping pretty well to different strains or different species that we weren't able to previously suss out using short read methods. So there's a lot of questions next of like, well, what does this mean and how we're implementing this? We're doing a lot of work where we're trying to look at individual strains, and this is allowing us to be able to track those strains a lot better, both the ICON PCR and the long reads Kinnex methods. We're also looking at a lot of different other types of ecological questions and different environmental questions. But for this purpose, I think that, the things that we realized based on going through and learning and working with the Kinnex platform before we used the autonormalization techniques, and then after, is that no matter what, this 16S gene is susceptible to PCR introduced residual error. And this is more compounded the fact that we're sequencing something that is 4 to 5 times bigger than what we've previously been sequencing. Within the ICON PCR platform, one of the things that we did recognize that we have to handle is we do need to control for primer dimerization for effective autonormalization. The primers that are published in the PacBio 16S, I think, work 16S protocol. That's pretty much what we've implemented. Exactly what we've implemented in all of our approaches. But we did notice that there is that polymerase, specifically the type of polymerase you use, can have inadvertent specific barcode induced dimer. We're not quite sure what is driving this, but I've had people email me with questions of like how have we mitigated this, and we're primarily using one polymerase that works particularly well in our applications, which is NDBQ5. We've also are trying to adapt this to. We've also are working on adapting some of the icon PCR approaches to other amplicon targets, like Jeremy mentioned earlier, whether that's ITS or operon based. That is where a lot of our methods in the lab are working towards now. Is how do we take these other targets and utilize them for using this autonormalization approach because we think it's particularly powerful. Further thoughts for at least for some tax, at least for the tax that I work with primarily, this intergenomic polymorphisms can basically help us distinguish some really difficult to parse taxa. There's a lot of things that we need to still resolve along those lines. I've been really like I think that I've been really convinced that this full-length 16S using this MassSeq platform is basically taking over all the work that we were used to using short read sequencing for because it is cost effective. And at least our hands, it is improving microbiome characterizations. And what I think is particularly exciting is that this is giving us a better intermediary to be able to say, yeah, we want to be able to sequence or do some shotgun metagenomic sequencing on those specific sample types. This allows us to kind of tell us yes, or point us to that direction. Right now, we're trying to couple this with some mapping to some genomes that we've obtained through hi-fi sequencing, like the hi-fi 16S connects with the with the genomes. And yeah, and lastly, like I said, we're trying to see how many things we can use this Kinnex platform for. So ITS is the current approach, but we're also working on some of the operon, like some of the full-length operon based methods. With that, I have a lot of people to thank. So this is fun. This work was funded by two of our research projects involving in our research unit. I also would be remiss not to acknowledge that a lot of the computational work was done using ARS resources and implemented through Samba. And then this work was collaborative between my research unit in the Tropical Pest Genetics and Mycology Research Unit, the Tropical Crop and Commodity TCCPR, which is Tropical Crop Commodity Protection Research Unit, and the Carl Hayden Bee Research Center. So with that, I'm happy to end my talk and thank you for attending and happy to answer any questions.
Awesome. Thank you very much, Charlie. Now we get to move into our Q&A. Our first question is pretty technical. It gets a little bit into the autonormalization weeds. So I'd like to first ask Mary to just recap for a second. What are the different modes for autonorm? And then we'll come back to the direct question about the best one to use.
Absolutely. Thanks, Jenna. So as we mentioned, AutoNorm is a software that's enabled in the Icon PCR, but there are four different modes you can choose from to enable auto normalization. So the four modes that we have are called slope, base X, base plus, and target fluorescence. Slope basically looks at as the name implies, the slope of your PCR amplification curve as it's progressing. At some point, it will hit an inflection point, which is where the rate of fluorescence increase is at its highest. Once it identifies that specific inflection point, the amplification will stop for that specific sample. Base X and base plus are very similar. As the name implies, the base part of it refers to the baseline fluorescence signal associated with each of your samples. So some samples may have a higher or lower baseline fluorescence, and it counts for that baseline level and sets a threshold above that baseline, above which your samples will stop. So for base X, that threshold will be a multiplier above the baseline, while for base plus, that is an additive value above the baseline. And then lastly, target fluorescence is, as the name implies, you set a specific fluorescence level, and once your sample crosses that fluorescence level, they will stop amplifying.
Perfect. Thank you so much, Mary. So now that we have that additional context, the question is around using base plus or base X for 16S microbiome amplicon library prep. So the issue is that there's variability in the baseline fluorescence, and obviously it's important to have that consistent baseline fluorescence to take that base plus or base X approach. So question for you, Charlie, which approach are you using, and do you have recommendations for how to handle that baseline fluorescence variation? The concern is that the slope method might be a little bit too much amplification for the 16S amplicons.
Yeah, so we use primarily slope, at least for most of our 16S work. We have messed around a little bit with the target RFUs too, but we really haven't done, at least for our lab, we haven't really done the like base X or base plus at this point. Yeah, I think that's basically our like we've settled it on I understand the concern, but we settled it on slope, and that seems to be the adaptive nature of it really helped us mitigate some of those issues, and like all the data that I showed today is all using slope.
Mary, any additional technical recommendations?
Yeah, absolutely. So for this specific application, so slope by nature does have to wait until the inflection point is attained. So in cases like this, it may be too much amplification present in order in getting your specific libraries. For minimizing any kind of amplification, but also taking into account variable baseline fluorescence, base plus is probably going to be your best choice here. Um, this way you can finally attune the level of thresholding above the baseline signal. Generally, base plus is better than base X because that additive nature really helps control the level of fluorescence addition as opposed to compounding it by a multiplier effect. So usually this would be something as simple as base plus 500 fluorescence units. But of course, the level of thresholding will be very specific on your assay and some of the initial data that you see with respect to your samples.
Awesome. Thank you, Mary. Another really interesting technical question that I'll field to Charlie first, and that is, do you set a max cycles to avoid over amplification artifacts? And if so, what do you use as a max?
So we use 30 for a max or 16S, mostly because of a lot of what we work with are they are just by their nature lower biomass. Like if we're working with soils, it could probably be a lot like exclusively soil or exclusively fecal. That could probably be a lot lower, and that'd be like you're cutting down an hour of your run. But for us, yeah, we use 30 because we have some samples that we just know are lower biomass. I will also say, like, we do run, I know for some of the fungal work where we're doing, where we're using ITS, we are running those a little bit lower. Those ones are, I think, 25 cycles. And that's mostly because we know that dimer starts forming at around cycle 27, at least under our conditions.
Mary, anything to add in terms of what you've seen from others?
Mhm. So typically when folks are running AutoNorm, they usually stick to the recommended cycle number already suggested by their protocols. Usually if they know they have some lagging samples, we typically add like 3 cycles above what the recommended protocol states. So that way it gives time for lagging samples to amplify. In general, whenever you're running any kind of AutoNorm run, you always will set a maximum number of cycles. So that way it allows your PCR run to stop eventually.
All right. Next question is a callback to the first one, but also a good transition to talking about sample type and sample variability. Any idea, thoughts about what might be driving the variability in baseline fluorescence that this is Stefan Green is asking that he's seeing in his 16S samples?
Um, it's a good question. So I'm just thinking about some of the samples that we have. Because of what we handle different we handle lots of different types of things. We have noticed that I think at least on our hands, we've noticed that some just higher titer or higher biomass, like we had a really effective extraction, and someone didn't do any QA/QC because we're doing it all on we're doing everything on 96 well plates, and we're just blindly transferring things over, and you see, oh, that one spiked. We usually go back and we'll check those samples and be like, yeah, those were really, really high, or those ones were having a higher level of starting DNA concentration. That's at least what I suspect maybe it would be without having any further knowledge of it. But that's also just how I know what like our lab and our people have noticed, like seen, like, yeah, this one sample looks a little bit off, and it ends up usually being concentration based.
All right. Mary, anything to add?
Yes, thank you. Yeah, to agree with Charlie, basically, sometimes we do see variation in baseline fluorescence just because of the input material going into PCR. So if you have samples that have higher, something with 100 nanograms of input versus something with 1 nanogram of input, it's very possible the 100 nanogram input sample will just give off a higher baseline fluorescence relative to the lower input samples.
So sticking with that variable input material, obviously, Charlie, you guys are dealing with a huge range. Is degradation something that you have encountered in your workflows? Have you dealt with degraded samples?
As in like I guess saying as in like purposely degraded, or as in like older samples.
Like older samples, yeah, or maybe, I guess you guys are dealing with exoskeletons and things like that. So I don't know, maybe too harsh of extraction.
Yeah, I mean, I think we haven't, not explicitly, I would say that like in terms of degradation, I mean, we've dealt with, I'd say, less less than ideal samples that were probably stored in less than ideal situations. But I don't think we haven't done anything systemic where we like, this is a sample that was like, we're not like, it wasn't like something that was from a museum or anything, like I would say, like that, where it's exceptionally challenging to extract DNA from. At
Gotcha.
least not my group. Other groups here, yeah, but not we haven't not in this. I say not in this application.
Got it. I guess, Mary, other applications where people have seen that, and have they found that they've had to run those degraded samples separately or deal with them differently?
It's a great question. So as one would expect, degraded samples might end up, you have less amplifiable targets to work off of. And usually, if you're trying to run a degraded sample side by side with a good quality sample, you'll find that you have different cycle numbers associated with each. Oftentimes, the degraded sample requiring more cycles to reach the same level of amplification relative to a good quality sample. With regards to running those samples on the Icon PCR, you really don't need to do anything much different. You can basically run both samples side by side. You set a high enough cycle number to allow for the lagging sample to amplify, and AutoNorm will basically take care of stopping your samples once it reaches the right level of amplification. So not only does it allow you to run, lower quality and high quality samples in the same run, but also the same applies to low input to high input samples. All of it can be applied and comfortably handled by the Icon, PCR, and AutoNorm functions.
So in terms of optimizing your samples for long read sequencing specifically, Jeremy, do you have recommendations for what extraction kits folks should use?
Yeah, so the commercially available kits from like Zymo, so like their mag bead extraction kits are really good, or mini prep kits. Any of their microbiome extraction kits yield pretty good sized fragments for either these amplicon approaches or shotgun metagenomics. Same goes for Qiagen with their power suite of extraction kits like soil, water, fecal, as well as their QIAMP for host depletion. And then there's been some recent studies that have come out, particularly from like Rob Knight's lab, where they found that the MagMax microbiome kit actually works really well as well. But Charlie can comment on what they've used in theirs.
Yeah, we pretty much at this point use basic Zymos. Yeah, we primarily their MagB kits primarily. I think we've made some modification. We do some modifications upstream for lysing, just because we want to minimize minimize any sort of cross-contamination in our preps. So it's a little bit more laborious on that front. But yeah, we primarily use like the mag beads implemented on a Kingfisher system.
Any recommendations from either of you specifically around what primer pairs to use for complex communities in soil?
So we use, I can tell you what we're doing now, and this is something that is not published yet. So we're using basically we'll use a 16S based approach for like this in this Kinix platform. But what's exciting to us is that we're starting to use the ITS 1 and 4. So not the full operon, but at least we're getting the full length ITS for fungal amplification, which is much you can't get, which is we're excited about because we're getting a lot of pretty good data with that. We implement those on the Kinix platform too, but what's been powerful for us is that we have them, and I think I mentioned this to Jeremy a couple weeks ago, is we just pull them all together, make one Kinix platform, and we barcode them separately, or barcoded separately, so we can make one big Kinix library, mixed Kinix library containing both, and sequence on the same cell. And so that basically lets us be able to kind of hit like on the same project plan. We're able to kind of get both fungal and bacterial ends from that, at least for the amplicon based work. I think Jeremy you might be able to comment on on long read based metagenomics, but I'm a little bit outside what we're doing currently.
Yeah, that's basically what my answer would be. And there's several labs doing that now where they'll do the 16S amplicon and then the paired with the ITS amplicon for fungal and the same connects reactions, and it works really well. The advantage of that too is it shortens the total fragment length too, so it increases the Q scores in addition to that. So it helps with accuracy and denoising.
This is a fantastic discussion. We do still have some more questions coming in. If we don't get to your question, don't worry, we will absolutely follow up. You can also reach out directly to Mary or Jeremy on the email addresses shown here. I just want to ask one more big picture question to send us out the door. Charlie, now that you've validated these workflows, how has your day-to-day general operations changed for most of your sequencing workflows? Are you still usually doing that manual qubit quant and the bead-based normalization? Are you skipping it entirely? Does it depend on the workflow?
It's skipped entirely. So usually for us, it's once we have for 16S, once we have an extraction done, we are at least for certain for sample type, mostly for insect sample types, we're blindly going extraction right into PCR, and then PCR right into pooling, and then right into four different plates or four different 96-well plates, and that gets pooled into one Kinix library. So it's just one workflow all kind of coming in together without really having to do a lot of manual curation for about 90% of what we work with on a day-to-day basis. There are 10% of them that are a little tricky, but that's kind of to be expected when you're working with heterogeneous samples.
That's amazing. What a way to end a webinar. Thank you so much again, Charlie, Jeremy, and Mary. And thank you, all of you, for joining us today. Again, you'll get a follow-up from us in a few days with the webinar recording. Have a great rest of your day.